Hello, in this podcast we're going to be looking at the process of going from DNA to RNA to protein. So we're looking at the mechanics of how genes are expressed. The genes are found in DNA and the expression is basically through the proteins. And we need another way of looking at the details here. Our objectives are to understand the processes of transcription and translation. First, we'll do a brief review of base pairing in DNA. DNA, remember, is a double-stranded helix like you see here. DNA has 4 bases: adenine, thymine, cytosine, and guanine, also known as A, T, C, and G for short. The adenine bonds only with thymine, thymine only with adenine. Cytosine binds only with guanine, and guanine only with cytosine. Now, RNA is another nucleic acid. RNA stands for ribonucleic acid. And there's some different structural differences between DNA and RNA is that in DNA has a sugar, and so does RNA. Both of them are made of nucleotides, which are a sugar, a phosphate, and a base. And in DNA, the sugar is deoxyribose, and in RNA, the sugar is ribose. Difference between the two is just one oxygen atom, and that can make all the difference. Another difference is that the 3 of the bases in RNA are the same as in DNA, but one of them is different. RNA does not have thymine, instead it has uracil. And basically you just have to substitute a U for a T when you're thinking of RNA. So if you have a DNA with an adenine and it's bonding to an RNA, it doesn't have a T to bind with, instead it binds to a U. Now, same thing, like if you have a thymine in DNA, then don't have to worry too much because that will bind with an adenine in RNA. And with C and G, it's the same thing. So the base couples, in DNA it's A and T, and in RNA it's A and U. And C and G is the same in both RNA and DNA. There are 3 types of RNA. One type is messenger RNA, or mRNA, and it's a copy of only one gene, and it's made from a DNA template, and that process is called transcription, and that we're going to focus on in this podcast. And then that messenger RNA itself is a template for making a protein, and that process is called translation. Another name for transcription is also called RNA synthesis, and another name for translation is protein synthesis. A second type of RNA is rRNA, also called ribosomal RNA, and that's basically just part of the ribosome. Ribosome is a combination of RNA and protein. The third type of RNA is tRNA, called transfer RNA. I would personally prefer what's called translate RNA because I think that sort of goes in with the translate, because the tRNA is what's involved in translating the genetic code over to the protein code. And we'll see that it helps assemble the protein by reading the mRNA. First, we'll talk about transcription, and I'm going to show you a clip about what generally is thought as a transcription or a transcript. If you watch many shows like Oprah, then you'll see that at the end they can provide you with a transcript. What's a transcript? You'll see in this clip.
And then you do the show. We send off for the transcripts, and then we give them to our stagehands, Pat and Kenny, and they read the Oprah transcripts for you. That's what we're going to do right now. Take it away, boys. Here we go.
She's sold 100 million records around the world. Please welcome one of the most popular singers, the fabulous Celine Dion.
Thank you, Oprah.
I understand you're taking some time off to meet life and create balance.
I don't want to go on stage and not be honest. I have other parts. I have other feelings. I have other dreams.
Go, girl. But music will always keep your spirit soaring, Celine?
Oprah, I want to sing and sing and sing and sing. I want to connect with the people. They fill me with joy.
This girl is talking the talk today.
Music is a way to express myself. It can help me cry when I need to let it out.
I could weep just thinking about it.
If you'd like to order a written transcript of Pat and Kenny read Oprah transcripts, write to us at 1697 Broadway, New York, New York, 10019.
To keep with the analogy, DNA replication, or replication, is sort of like if you had set your DVR to record an episode of Oprah. And then you can watch it anytime, and it's exactly the same thing as if you had watched it when it was first broadcast. Transcript, on the other hand, is just simply like it's basically just written down what's there. It's missing the information in the same language, but the format's different, and some things are missing. And it's sort of the same thing with transcription. In transcription, you've got one enzyme called RNA polymerase. What it does, it comes in and it unzips the DNA at the gene. So you don't get the whole DNA that's transcribed, it's just one gene. And it reads not both strands, but only one strand. So the DNA is double-stranded, mRNA is only single-stranded. So it basically looks at the genetic code in the template strand on DNA, and from that the RNA polymerase enzyme synthesizes or constructs the strand of mRNA. In class, you are going to be required to make an RNA strand given a DNA template. And basically you just have to remember that A binds with U, T binds with A, C binds with G, and G binds with C. So if we look here at, let's get the eraser up. So if we look over here, A binds with U, and then T binds with A. What does G bind with? C. There are a whole bunch of A's, and what do they all bind with? They all bind with U, not T. C then binds with G. What does A bind with? U. And of course A binds with U. G binds with C. The other G binds with C. T with A, not U. T binds with A. But A does not bind with T. A binds with U. C with G. And A with U. C with G. And I hope you're going along with me, figuring out before I tell you what it is. So A binds with U. T with A. C with G. T with A. It's getting a little off there. And A goes with U. Now after this is constructed, then the RNA is edited. Parts of the RNA are removed. The parts that are removed are called the introns. The parts that remain are called the exons. So you have a shorter strand, and that particular thing is called the mRNA. And then that will leave the nucleus where the DNA is, and will go out into the cytoplasm to find the ribosomes. Once it gets to the ribosome, then translation begins. With transcription, we had the same language. Translation, we're changing languages. We're going from the language of the bases, ATTC, to proteins, where we have the language of just a sequence of amino acids. This occurs in the ribosome. First thing that happens is that the mRNA attaches to the ribosome, and you see that strand is the mRNA, and the ribosome has two parts to it. After that, the tRNA comes in and attaches to the mRNA, and you see how one end will bind to the mRNA, and on the other end, the MET is an amino acid. And then a second tRNA comes in and attaches, and a bond forms between the two amino acids. And you see how it also has three base sequences that bind to three base sequences in the mRNA. These three base sequences are called codons. You have to know that word. And then the ribosome moves over, the empty tRNA is ejected, and it will eventually find another amino acid, and it can then be recycled. Then the next amino acid that matches the next codon comes in, and that amino acid then binds with the previous amino acid, and the process is then repeated, and it just continues until a stop codon is encountered, and that basically just stops the whole process, and it just detaches it, and you end up with your polypeptide, which is a long sequence of amino acids. Now here's how you find the amino acid from the codon. First thing we need to do here is, in this RNA strand, let's first identify the codons. So what you can do is just make a comma here, count of 1, 2, 3. Actually, it might be better to, I usually put commas in, but actually this might be good too. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. Now we only have 2 here. There should be a third over here. We'll find out why there isn't one soon. Okay, so you have to look at this codon chart, and if you ever have to solve this on a test, or on the Regents for that matter, you will be given this chart. You have to know how to use it. You don't have to memorize it. So first let's look at UAC. We go to our first base, that's a U. Second base, then we go over to the A, and then we find UAC, and you see TYR. So let's go over here in our protein. First amino acid is tyrosine. TYR, all the 22 amino acids are given 3-letter initials. UUU. So let's go to our first base of U, second base of U, third base of U, and we have phenylalanine, or PHE. And yes, sometimes in Regents exams you are expected to do this, and you may be expected to do this on one of my exams. Next one, UUG. So we go first base U, second base U, and then third base we got a different one in there, leucine. If you ever saw Jurassic Park, leucine is the amino acid which the dinosaurs weren't able to synthesize, which should have kept them from escaping, because leucine is amino acid you need to make proteins, and without leucine they can't survive. Okay, so that was UUG. Next one is UUC, and we're back to phenylalanine. CAU, so we go down to first base is C, second base is A, third base U, histamine, HIS. You do not need to know what the letters stand for. Just putting down HIS is enough. And now we're up to, that was UUC, so, the CAU. Next is GUG. So first base is G, second base is U, third base is G, and that is valine, VAL. UAG. First base is U, second base is A. That's why the AU is not a full codon. UAG is a stop codon. So that's basically saying this is where the protein stops, and basically protein synthesis stops here, and the ribosome disengages from both the budding protein and from the mRNA, and that's the end of the protein synthesis. So we don't have to worry about the AU at all. And as it's synthesized, the protein folds, and it becomes a 3-dimensional structure, and the shape determines the structure. What I'm showing here is how one slight change, one mutation, can make the whole difference. Because remember, watching that video, we saw they said there's one base change made a big difference, and that's what you see here. This top picture here is normal hemoglobin. That's the shape, and it has to be that shape in order to function perfectly. The shape you see below is caused by one base change, and that gives sickle cell anemia. And what happens is that if someone has only one copy of each, they're going to end up with both proteins, and that person will be basically resistant to malaria and will never come down with malaria, and that's why it's so common among Africans in sub-Saharan Africa. But if someone has 2 copies of the sickle cell allele of the sickle cell gene, then they're going to end up with both of those, and they're going to end up with sickle cell anemia, because in that shape the hemoglobin does not carry oxygen as well as in the normal shape. Now here we have the central dogma, which is DNA to RNA to protein, and this in this diagram here, I just have here to basically summarize for you the whole process of going from DNA, and you see DNA can replicate to make more DNA, and then a part of the DNA can be transcribed into RNA, and the RNA is then transcribed into making the protein. Now we come to the concluding questions. First one, what is the difference between DNA and RNA? Number 2, where does transcription occur and what is made? Number 3, why is it called transcription and not replication? Number 4, where does translation occur and what is made? Number 5, what is the role of tRNA? That concludes this podcast, and I'll see you in class tomorrow.